Autonomic nervous system. So what's the effect of drug on the nervous system? In this lesson, we will tackle the sympathetic system, adrenergic response, parasympathetic system, and cholinergic response. First, what is autonomic nervous system? It is sometimes called the involuntary or visceral nervous system because it's mostly function with a person having little conscious awareness of its activity. Working closely with the endocrine system, the ANS helps to regulate and integrate the body's internal functions within a relatively narrow range of normal on a minute-to-minute basis. The ANS integrates parts of the central nervous system and peripheral nervous system to automatically react to changes in the internal and external environment. What's the difference between central and peripheral nervous system? Central nervous system consists of the brain and spinal cord. The brain plays as a central role in the control of most bodily functions, including awareness, movements, sensations, thoughts, speech, and memory. Some reflex movement can occur via spinal cord pathways without the participation of brain structures, while the peripheral nervous system is a division of the nervous system containing all the nerves that lie outside the central nervous system. Primary role of the PNS is to connect the CNS to the organs, limbs, and skin. What's the structure and function of the autonomic nervous system? The main nerve centers for the ANS are located in the hypothalamus, the medulla, and the spinal cord. Nerve impulses that arise in peripheral structures are carried to these centers by afferent nerve fibers. These integrating centers in the CNS respond by sending out afferent impulses along the autonomic nerve pathways. These impulses adjust and functions of various internal organs in ways to keep the body's internal environment constant or homeostatic. Moving on to the sympathetic nervous system. It is sometimes referred to as fight or flight system, or the system responsible for preparing the body to respond to stress. Stress can either internal, such as cell injury or death, or external, such as perceived or learned reaction to various external situation or stimulation. For the most part, the SNS acts much like an accelerator speeding things up for action. Adrenergic response. Sympathetic postganglionic nerves that synthesize, store, and release norepinephrine are referred to as adrenergic nerves. Adrenergic nerves are also found within the CNS. The chromaffin cells of the adrenal medulla also are adrenergic because they synthesize, store, and release norepinephrine as well as epinephrine. Norepinephrine synthesis and storage. Norepinephrine belongs to a group of structurally related chemicals called catecholamines that also includes dopamine, serotonin, and epinephrine. Norepinephrine is made by the cells using tyrosine, which is obtained in the death. Norepinephrine release. When the nerve is stimulated, the action potential travels down the nerve axon and arrives at the axon terminal. The action potential then depolarizes the axon membrane. This action allows calcium into the nerve, causing the membrane to contract and the storage vesicles to fuse with the cell membrane, releasing their load of epinephrine into the synaptic cleft or cleft. Adrenergic receptor. It can be stimulated by the neurotransmitter released from the axon in the immediate vicinity, and they can be further stimulated by circulating norepinephrine and epinephrine, secreted directly into the bloodstream by the adrenal medulla. The receptor sites that react with the neurotransmitter at adrenergic sites have been classified as alpha and beta receptors. Termination response. Once norepinephrine has been released into the synaptic cleft, stimulation of the receptor site is terminated and disposal of any extra norepinephrine as well as the neurotransmitter that has reacted with the receptor site must occur. Most of free norepinephrine molecules taken up by the nerve terminal that release them in the process called reuptake. For the parasympathetic nervous system, first, it increases motility and secretion in the GI tract to promote digestion and absorption of nutrients. It also decreases heart rate, contractility to conserve energy and provide rest for the heart. And lastly, constriction of bronchi with increased secretion. For the cholinergic response, neurons that use ACH as their neurotransmitter are called cholinergic neurons. These are four basic kinds of cholinergic nerves. First, all preganglionic nerves in the ANS, both sympathetic and parasympathetic. Postganglionic nerves of the parasympathetic system and a few SNS nerves such as those that re-enter the spinal cord and cause general body reactions such as sweating. Third, motor nerves on skeletal muscles. And fourth, cholinergic nerves within the CNS. For the acetylcholine synthesis and storage, ACH is an ester of acetic acid and an organic alcohol called choline. Cholinergic nerves use choline, obtained in diet, to produce ACH. The last step in the production of the neurotransmitter involves choline, acetyltransferase, an enzyme that is also produced within the cholinergic nerves. Just like norepinephrine, the ACH is produced in the nerves and travels to the end of the axons, where it is packaged into the vesicle. To be a cholinergic nerve, the nerve must contain all the enzymes and building blocks necessary to produce ACH. Then, for the acetylcholine release, the vesicles full of ACH move to the nerve membrane. When an action potential reaches the nerve terminal, calcium entering the cell causes the membrane to contract and secrete the neurotransmitter into the synaptic cleft. The ACH travels across the synaptic cleft and reacts with very specific cholinergic receptor sites on the effector cell. For the cholinergic receptors or ACH receptors are found on organs and muscles. They have been classified as muscarinic receptors and nicotinic receptors. The classification is based on very early research of the ANS that use muscarine and nicotine to study the actions of the parasympathetic system. And lastly, for the termination of response, once the effector cell has been stimulated by ACH, stimulation of the receptor site must be terminated and destruction of any ACH must occur. The destruction of ACH is carried out by enzyme acetylcholinesterase. This enzyme reacts with the ACH to form chemically inactive compound. The breakdown of the released ACH is accomplished 1/1000 seconds and thus receptor is vacated, allowing the effector membrane to repolarize and be ready for the next stimulation.